Thank you for the kind introduction. I just wanted to mention that for this live biotherapeutic products, there was a monograph or general monograph set up for the EDQM for the European Pharmacopoeia, and I had the possibility to join that group. They already had started their work, so I joined a bit later, but yeah, was in the finalization process involved for this specific general monograph. I will come to that later, just to let you know how yeah, things are yeah, set up and built in and to reflect the requirements that are already in place for those types of products. Okay, thank you very much for the kind introduction, and welcome back after the coffee break. I hope you enjoyed. And now we will come to live biotherapeutic products. And just to start with the definition, what are live biotherapeutic products? They are medicinal products containing live microorganisms as active substance. And examples are a mixture of gut bacteria like Enterococcus species, E. coli subspecies, lactobacilli or bifidobacteria. These are yeah, available as food additives or also as drugs. And there are also lactobacilli for vaginal administration. Some of them were formerly marketed in the EU as medical devices under the former legislation, under the Medical Device Directive, but are now considered a medicinal product, meaning they need approval for yeah, to be able to market them. So this is now the first slide, or as I mentioned, those products can be regulated differently, and I would like to know from you how they are regulated in your markets at home.
Katrin, your question is posing lots of murmuring. There's lots of murmuring going on in the room as people trying to work out the answer to this one. It's quite interesting.
Okay, yeah, I see. Is it the number here above the?
So we have 67 responses so far out of there are 118 people online.
Okay.
Plus we have all the
Okay.
colleagues in the room, so we're not quite there. We ought to wait just a few more responses.
And can I see the results? Is it possible to make them available after a certain time?
Yeah, you should be able to. You should be able to see them on screen. But currently, at the moment, we have 62%, 63% saying both, 26% saying drugs. Of course, it's now just changed, and 14% saying as a food additive.
Okay.
But the overwhelming majority is both.
Both. Okay.
Although just nudging up to 30% for drugs. So I'm going to do the chairman's prerogative here. I'm going to ask around the room: has everybody answered? Does anybody still want time to answer, or are we good to go move on? Okay, someone just shouted out "move on." Very good. Okay, right. So Katrin, to you. There you have your answer. So the answer is 62% both, 28% drugs, and 14% food additives.
Okay, thank you very much for this responses. I think it's quite similar to the situation in the EU. May I now move my slides again? Ah, no, this is the second question. Okay. have you ever evaluated live biotherapeutic products? And this is obviously mostly the case if they are regulated as drugs. So this would also be an interesting question for me.
Again, Katrin, quite a lot of murmuring in the room, which is good. But it seems to be about 65% have answered no.
Okay.
We're still about 75 people have responded, so we. I think we're good.
It's the.
Yeah, I think it looks as people in the room have answered, so I think that's your answer. 65% no, 35% yes.
Okay. Yeah, thank you for this. And yeah, this is well in line with the responses regarding how it's regulated. So it's yeah, in most cases, I expect that you will only review them if you if they are regulated as drugs. So what are the differences for food additives and drugs in terms of regulation? So food additives belong to the foodstuff, and they serve to supplement the diet of healthy persons, whereas drugs are intended to cure, alleviate, prevent, or detect diseases, ailments, bodily injuries, or pathological complaints, or this, yeah, let's say the general definition of a drug. So the difference is that a food additive is intended for use for healthy persons, whereas LBP as a drug are intended to cure something. Food additives, this is quite obvious, are subject to food law, whereas drugs are subject to medicinal products law, and related to this, also to the Medicinal Products Act, to the in Germany to the national law, and of course they are also subject to Pharmacopoeias and the respective requirements laid down there. Food additives in Germany must be notified to the BWL, which is the national authority for foodstuff in Germany. And drugs have to be approved by a national competent authority, for example, the BfArM. For food additives, it's not necessary to prove efficacy or safety, and the manufacturer is responsible for the safety of the product, but it's not not needed to prove the safety in vis-a-vis an authority. So yeah, this is a different concept. And for drugs, the situation is different. The manufacturers must submit clinical studies in the authorization procedure that prove the efficacy and safety of the medicinal product. Of course, in the end, the medicinal product manufacturer is responsible for the safety of their own product, but they have to prove efficacy and safety to get approval for their product to access the market. Regarding food additives, it's usually possible to deviate up to 50% from the label claim regarding the quantity of components of the food additives, whereas the quantitative information on the packaging for drugs may deviate by no more than 5%, usually at least, from the actual dosage of the active ingredients. The situation is quite different for LBP. I will come to that later because of the fact that the number of viable cells is not to be stated as precisely as let's say the amount of a chemically defined active ingredient in a finished dosage form. For food additives, no maximum quantities are specified for ingredients except for technological additives. And for drugs, the dosage of all ingredients are checked and precisely defined as part of the authorization procedure. They are not necessarily stated on the label. the amount of only of the active ingredient is clearly defined, but yeah, to get approval, the exact composition has to be stated in the respective documentation. So coming back to the LBP, live biotherapeutic products, these were first defined in the European Pharmacopoeia to write a general monograph on those. And I will do now a slight excursion on the European Pharmacopoeia, and I'm sure quite a number of you will be very familiar with that with European Pharmacopoeia. But yeah, just to further explain, yeah, what's the meaning of a general monograph, and how does that fit into the overall structure and idea of European Pharmacopoeia? Perhaps I should state in advance that the European Pharmacopoeia is reflective of approved finished products and approved medicines. It's not inventing standards for things that have not been approved in any area of the yeah, where the European Pharmacopoeia is in force. It's reflective of already approved drug products, so it's not possible to define standards for something that may be done in the future, something new that may be done in the future. It's approved products form the basis for setting up monographs and also general monographs. So the tasks of the European Pharmacopoeia are to promote the health of the population by providing generally applicable rules and standards for all those involved in the healthcare system. It's also set up to ensure the safe use of medicinal products and to ensure the free movement of medicinal products in Europe. And it's intended to ensure the quality of medicinal products on the European market. So the the EP standards are recognized worldwide, and is the European Pharmacopoeia is the official pharmacopoeia for entire Europe, and legally binding standards for all medicinal products in Europe. For example, raw materials, preparations, and dosage forms must comply with the respective requirements if they exist. Very famous standard is the standards for the excipients used in finished products. They most of them are monographed in the European Pharmacopoeia, and then they have to comply with those standards. Perhaps it's also worth mentioning that the European Pharmacopoeia describes the minimum requirements and for the respective materials and active substances and so on. Usually additional specifications are set, at least for active substances. The EDQM harmonizes the specifications for substances from various sources and establishes transparent monographs with an impurity profile. And specifications and valid analytical working methods are developed and laid down in the respective monographs. And EDQM also provides reference substances that can be used or that should be used in the context with the respective analytical methods. Monographs and other texts are prepared in such a way that they meet the expectations of, on the one hand, the regulatory authorities, but also of those who are responsible for the quality control of the medicinal products and their ingredients, meaning, for example, the qualified persons in the manufacturing, the manufacturers of the drug products, and also of the manufacturers of starting materials and finished medicinal products. So we have several sections in the European Pharmacopoeia, and this is an overview of the general structure. First of all, the introduction. Then we have the general notices, and it's worth reading them because they are applicable to all monographs and texts of the European Pharmacopoeia, and they contain instructions for understanding texts and conventional expressions and so on. And yeah, it's a general reading before starting to use general chapters and monographs. Then we have the general chapters. They are describing in chapter 2 the methods of analysis. In chapter 3, materials for containers and also containers for pharmaceutical use. Chapter 4, reagents, and chapter 5 contains general texts. The methods of analysis in chapter 2 are stated there to avoid repetition in the individual monographs and texts. And they are subdivided into the principle of the method, the equipment and apparatus needed to execute the method, requirements for equipment performance, how to prove that performance, how to execute performance qualification, and also very important, it describes the procedure itself and validation requirements for the procedures. For example, 2.2.27 thin layer chromatography, sorry for that, and 2.2.29 HPLC, high performance liquid chromatography, and therein is a cross-reference to 2.2.46 chromatographic separation methods. This describes how far parameters may be changed without requiring revalidation, and this is also an important part of the description. 2.6.12 is on microbiological examination of non-sterile products. So these are just some quite famous examples, I would say. In chapter 4, reagents are described, which are needed for the specific analytical procedures. And reagents used in tests have the suffix R. And the purpose of section 4 is to assist in the selection of appropriate reagents. So there are quality criteria or properties of the reagents described, but this description is not exhaustive. And in the general text chapter, we have, for example, a text on microbiologies. We have texts on biological test methods. We have a text on statistical evaluation of biological test methods. And I copied here, for example, 5.1.6 alternative methods for control of microbiological quality, and so on and so forth. So you find a lot of information on these general considerations or general tests. Then it comes to the general monographs and dosage form monographs, which is the next section of the Pharmacopoeia, containing information on pharmaceutical preparations as such, on radioactive drugs, on parenterals, on chemical precursors for radioactive drugs, and so on. So, for example, we have general monographs for substance classes, like substances for pharmaceutical use, like pharmaceutical preparations, radioactive drugs, monoclonal antibodies, and also the live biotherapeutic products. And we have also general monographs on dosage forms, on parenteral preparations, capsules, tablets, and so on, describing the general requirements for those. The purpose of these general monographs is to cover aspects that are not covered in individual monographs, but are applicable to the substances and preparation of the class or dosage form concerned. And they are applicable to substances and preparations covered by the definition, unless it is stated in the text that certain substances or preparations are excluded. And they are complementary to individual monographs. And they are also applicable to substances and preparations for which there is no single monograph. And this is the case for the LBP. We have only this general monograph here describing the overall requirements for such types of products. And we don't have individual monographs for individual products here. And then just to complete the overview, we have the individual monographs, which are more than 2,500 individual monographs, for example, chemical substances, herbal drugs, antibiotics, vaccines, and so on. So now I come to the general monograph on live biotherapeutic products. It starts with the definition of LBP, like it's always starting the monograph with the definition of what is described. So they are medicinal products containing live microorganisms, which are bacteria or yeasts for human use. One strain or combination of several strains may be used in one medicinal product. So you may combine more than one strain in the final product. And their administration is orally or vaginally. This reflects what is currently approved in the regions where the European Pharmacopoeia is valid. We have different pharmaceutical forms. They may be capsules or solutions or suspensions, not solutions, suspensions. So it's not prescribed exactly what kind of pharmaceutical form it is. And the strength of the final product is defined by counting the viable and culturable microorganisms. And fecal microbiota transplantation is excluded. This is something else, but this is not so. It's not defined on the strain level and so on. So it's a totally different approach, and they are not in scope of the LBP monograph. The aim of the monograph is the definition of quality requirements to guarantee that the product is comparable to those batches that were shown to be safe and effective in the clinic. You remember, for drug product approval, it's necessary to show that, and in contrast to the food additives. So we have clinical studies forming the basis for approval of those types of products, and the bio batches used in the clinic, they have been shown to be safe and effective, and the manufacturing process and control of the LBP needs to be defined in a way that it's representative of the batches that were used in the clinic. So we need consistent quality, and for this we need a proper definition of the production process. So the general provisions on the production methods are, as I said, we need a consistent process to yield consistently an LBP comparable with the batches of proven clinical efficacy and safety in man. This is the text taken from the monograph. And what is needed to achieve at a consistent process. We need process parameters appropriately defined, and in-process tests also, with defined limits and or ranges according to the process that was used for the clinical trial material. And also, we need a classification of the process parameters and in-process tests with regard to their criticality, and this needs to be adequately reflected in the section S2. S2.2 for the description and S2.4 for the critical process parameters and controls in process tests. In the Pharmacopoeia, it's very important that a consistent wording is used. Therefore, we are still talking about a seed lot system as basis for consistent production. From the biologics world, you may be more familiar with the terminology of cell banking systems, but the meaning is the same. Excipients may be added to the LBP, and the viral safety general text 517 should be considered as applicable once, for example, raw materials of human or animal origin are used during the cultivation of the microorganisms. The monograph also describes how the microorganisms used need to be characterized. The characterization should be done on the strain level, on the master seed lot, which is correspondent to the master cell bank. So it's the origin of strain that should be described. Where was it isolated from? And yeah, perhaps it's also deposited in a strain depository, in an official one, and has assigned a strain number, an official strain number. Then if the strain has been manipulated, this should be described as well. Then we need a phenotypic and genotypic characterization of the strain. That means on the phenotypic side, macroscopic and microscopic description, biochemical tests, and so on and so forth. On the genotypic characterization, molecular genetic tests, sequencing as applicable, and also we need information on the stability in terms of phenotype and genotype. Ideally, the strain does not change during cultivation. Yeah, this is for the sake of consistent quality of the final product. We need stability in this respect. And the strain needs also to be characterized with respect to the presence or absence of antimicrobial resistance genes. This is really an important one. And if those are present, which poses a safety risk in the end, transferability of such resistant genes needs to be evaluated. And also the presence of virulence factors needs to be evaluated, because those may have an impact on safety of the final product. For orally administered strains, the survivability in gastric acid and bile needs to be demonstrated to somehow get an idea of how much of the cells will arrive in the gut still alive. Then we have a seed lot system, which means a two-tiered cell banking system. We have a master seed lot and a working seed lot established. And this is needed also to ensure consistency of the process, of the manufacturing process. And establishment and testing of a new working seed lot should be described to avoid variations in future. This is the same story as for recombinant biologics, for example, when you have master cell bank and working cell bank, and you want to establish, the master cell bank is usually set up in a way that it will cover the entire life cycle of a product. but from the master cell bank, several working cell banks are usually derived throughout the product life cycle, and if a proper specification for those is included already in the CTD, then it's not necessary to file a variation each and every time a new WCB is or working seed lot is established. And if a new master seed lot is needed, if the master seed lot does not cover the entire life cycle of your product, then a full characterization of the new master seed lot is needed, and of course this triggers a variation procedure and has to be approved by the authorities. The manufacturing process needs to be revalidated once a new master working seed lot system is established. This makes only reference to the establishment of a completely new system, meaning a new master cell bank. As I said, variation needs to be filed and approved prior to use of the new master. Then we need also, it's also described in the LBP general monograph, an adequate definition of the culture media. With respect to the qualitative composition, usually we don't get the exact quantitative composition because this is very often confidential and proprietary to the media provider. But information on the qualitative composition is definitely needed to ensure that materials are avoided that could cause allergic or other undesirable reactions in humans. And if there are materials of human or animal origin, TSE and virus safety has to be proven by respective documentation. In contrast to, for example, recombinant proteins, the LBP manufacturing process does not really deplete the media from the final product, because there are usually no significant purification steps during the manufacturing of the active substance. That means that in the end, media components will be present in finished products at considerable amounts. Therefore, this is another reason why it's so important to get enough information on the culture media. Growth and harvest should be appropriately defined or described. Inoculation in a defined media, and then the scale up to the final fermentation. Purity is tested of the harvest by addressing the morphology of the cells using microscope. Also, microbial contamination has to be investigated, and there are references made to the analytical methods 2636 and 2638. I will come to this later. Oh no, not later, directly. Sorry for that. So why do we need special descriptions of determination of microbial purity? I copied here, sorry or apologies for this table being so tiny. Just focus here on the non-aqueous preparations for oral use as stated in the general text 514. We have this table here, and for non-aqueous preparations for oral use, TAMs and TIMs are defined to be present at a maximum of 10 to the 3 and 10 to the 2, I think, per milliliter, and specified microorganisms are stated in the last column here of the table, and for non-aqueous preparations for oral use, absence of E. coli has to be demonstrated, and also for aqueous preparations for oral use. And for vaginal use, the requirements are slightly different. We have here 10 to the 2 and 10 to the 2 for the TAMs and TIMs, and there are more specified microorganism or other specified microorganisms, the absence of which has to be demonstrated. That is Pseudomonas and Staphylococci, and also absence of Candida of fungi. So these are the requirements for, as stated in 514. But the problem is that the total aerobic microbial count and the total yeast and molds count is the way how this is counted is not really usable for LBP, because LBP as such already contain millions of microorganisms, and in the presence of those microorganisms it's nearly impossible to do the usual tests required to demonstrate compliance with these requirements here. And it also doesn't make sense to talk about total aerobic microbial count, TAMS. It's more sensible to talk about aerobic microbial contamination count, because usually we have a very, very high count of aerobic microorganisms, and the same is true for the yeast and mold count. So we need other methods for those types of products, and these are described in excuse me, I just spotted the typo here in the title. The first one I want to explain to you is the 2636, the microbiological examination of LBP and the test for enumeration of microbial contaminants. The problem, as I mentioned, is the growth inhibition of contaminants by LBP. For example, lactobacilli have a quite low pH environment, and by this they suppress the growth of other aerobic microorganisms. So we have to turn the situation around and select growth conditions for the test such that the growth of the LBP is inhibited. And the test should be able to detect microbial contamination in the presence of the LBP. And this needs to be established and demonstrated. And in 2636, a number of possibility is described for particular species which are currently used as LBP, but there is no one size fits all. So it's of course possible to modify the tests there, and a decision tree how to do that is included in the in 2636. So, for example, suitable media are stated for various types of LBP. For example, for lactic acid bacteria, sugar-free agar plates may be used, and here are also the conditions stated. And and here the decision tree starts with the question, is the enumeration method described in this chapter suitable for the LBP? And if yes, you can just follow the procedure, and if no, modification of the methods may be needed, and these may include increase in the volume of diluent or culture medium, or incorporate a neutralizing agent, surface active agent, or solubilizing agent into the diluent or medium to suppress the growth of the LBP, or a combination of both. then if you've done that, it should be verified if the method is now suitable in presence of the LBP. Is the growth of the contaminant still inhibited? Then further modifications should be investigated, and these may include the growth modification of the growth conditions like temperature, time, pH, etc., or use microorganism growth inhibitors or other suitable media. Or a combination of the above measures. And examples of approaches are given in the section 6 of this 2636. And then if you've changed your method, it needs to be validated by verifying appropriate parameters. And this is this is why this chapter is quite long. How to describe what steps can be applied and can be gone through to achieve a suitable method setup. Similar considerations apply for the tests for specified microorganisms described in 2638. Again, the ability of the test to detect the target microorganisms in the presence of the LBP to be tested must be established. So the test methods are described again, and also here we have a decision tree to be applied if the test does not work for a particular LBP. Then we come to the finished product. The final bulk or final lot may contain a strain or a mixture of 2 or more strains. Stabilizers and other excipients may be added depending on the dosage form. And if the final lot is an aqueous suspension, it is distributed into sterile tamper-evident containers, and the final lot complies with the requirements under identification tests and assay. Certain tests prescribed for the final lot may be carried out on the final bulk prior to filling, provided it has been demonstrated that subsequent manufacturing operations do not affect compliance. So this is quite common, I think, also for other products you may be familiar with, that several tests can already executed on the level of the bulk prior to filling. What tests need to be applied? At first, identification of the active components. If you have more than one strains, might be a bit more difficult. Suitable methods are used or needed. And the requirement here for suitability is that the methods are specific and discriminative in relation to other strains handled at the same site. This is something that may be applicable, in fact, to finished products which are manufactured at sites where more than one LBP is produced, and then specificity of the identification method should comprise or should be demonstrated in particular with respect to the other strains that are handled at the same site. And sometimes you have situations where more than one lactobacillus strain, for example, is handled at the same site. Or more than one lactobacillus strain is contained in a final product. So this might be quite tricky to demonstrate that both of them are present in the final product. Microbial contamination has to be shown according to 2636 and 38. Then pH is also an important test, and water, water needs to be addressed. Usually we have lyophilized, or not usually, we have usually lyophilized products, And for stability of those, the water content is really an important parameter, and this can be addressed by water content itself, loss on drying, or water activity. And then also very important, the assay, because the potency of the product or the dosing is done according to the potency, and potency of each strain is expressed in CFU per gram, per milliliter, or per unit. It may also be expressed in viable cells in agreement with the competent authority. So we have here again the acceptance criteria for microbial contamination. This is now the table for LBP, in contrast to the table for the non-LBP products. Taken this was, as I shown here. This is taken from the general text 514, and for the LBP we have specific requirements for the AMCC and the YMCC, and it's expressed in CFU per gram or milliliter, and yeah, CFU stands for colony forming unit, and yeah, the potency, which is different, it's also expressed in colony forming unit, but may also be expressed in viable cells. This was taken on board due to the fact that we have one product on the market where the where the potency is stated in viable cells and not in colony forming units. It's a matter of analytical procedure used to count those. So we see here for the AMCC the requirements for non-aqueous preparations for oral use. 10 to the 3 may be present, and 10 to the 2 YMCC may be present, and E. coli needs to be absent, except if you have a product that contains E. coli, of course. Then we have aqueous preparations for oral use, and there the numbers are lower because there is still growth, or the possibility that those contaminations may grow, and also again absence of E. coli. And for vaginal use, you have also the requirement for 10 to the 2 AMCC and 10 to the 1 for YMCC, and absence of, as I mentioned before, Candida, and the absence here of the specified microorganisms is similar to that stated in the 514 table. Labeling requirements, the name of the strain or the strains needs to be stated, route of administration needs to be stated, storage conditions and expiry date. The potency of each strain is expressed in CFU or in viable cells, and also the name of any stabilizers and other excipients should be stated on the label, but not the quantity. And where applicable, for freeze-dried preparations that need to be reconstituted prior to use, the name, composition and volume of the reconstituting liquid to be added, and the storage conditions and the period of time within which the LBP is stable after reconstitution. So now I come to the last part of my presentation on common deficiencies and case studies. What we see for such type of products upon approval procedures, review procedures, we see typical deficiencies mainly in the sections S22, S24, meaning that we often receive only an insufficient description of the manufacturing process. It's not defined at the appropriate level of detail, and critical process parameters are not included. For example, cell culture volume, exact conditions, pH, temperature, duration, and also criteria for the next step during inoculum build-up, for example, or criteria for harvest. Then sometimes the flow diagrams are too limited, not stating the controls and the process parameters, and also process validation is missing. And yeah, this applies to S2.5, of course. Then typical deficiencies in S2.3, the origin of strains in particular important if the reference is made to literature data. We sometimes receive applications according to paragraph 10a, which means well-established use, where for the clinical part reference is made to literature data, and it's yeah, and then for that purpose it's really needed that that the relationship between the strains that were used in the literature and that are used for this particular product, that this relationship is clear, and that the origin of the strains is adequately described. Then there's also missing characterization with respect to virulence factors and antimicrobial resistant genes. Yeah, this is quite a lot to do if such a deficiency is raised by the authorities. It's yeah, this takes quite some time for the applicants to respond to those questions. So this is just an example. I intended initially to have an additional Slido here, but this did not work. So we received such a submission for the freeze-dried pellets, which contain in the end the freeze-dried strain, and this was the specification proposal we received. It contains total cell count with a lower limit, and water activity, and total aerobic microbial count less than 2000 CFU per gram. And I would like to ask you if you think that this is sufficient.
So we don't have a Slido.
Perhaps you in the room?
So we don't have a Slido.
No,
So I think the way we're going to do it, Katrin, is show of hands. So perhaps a show of hands. Do we think this is sufficient? If you think that is yes, sufficient, please raise your hand. No hands so far, Katrin. So if you think it is not sufficient, raise your hand. We have a sea of hesitant hands saying no, it is not sufficient. Do we need a bit more time?
Yeah, okay.
We've actually got some interesting conversations going on. People are, give them a couple more minutes. Montenegro is conferring. Feels almost like Eurovision. Okay, Montenegro. What are you going for? Sufficient, insufficient? So Montenegro's voted no. Okay, nul point. Okay, so Katrin, the answer, it seems to be that the consensus here is that it is insufficient. And the answer is?
And yeah, the answer is no. And yeah, then the question is, if it's no, what is missing? And I propose here identity test, B, reference to analytical procedures, and C, reference for microbial examination tests. And yeah, I think this might be a bit more difficult to achieve just by raising hands or not. Yeah, I will tell you the results. So it's
well, Catherine, let's but perhaps just to, because I think this is actually an interesting exercise. It's part of the participation in the meeting. can we give, before you tell everybody the answer, can we just sort of get the groups to have a think about, turn to your neighbor on your left and your right and see if you can work out what the answer is. So we've agreed it is not sufficient, so what's missing? Is it the identity test, the reference, any reference to an appropriate analytical procedure, or reference to an appropriate microbial examination tests? So Catherine, you can't hear it, but there is murmuring going on, which is always a good sign.
That's good.
We'll give them just a couple of minutes. Yeah, thank you. And online, just because you're online and hidden from view doesn't mean that you also can't vote. So if any of you want to put in your reflections in the chat, you are very welcome to do so, please. Okay, that's gone. The room has gone suddenly quiet. I don't know why. So anybody want to take the microphone and just give their views on the answer to this, to Catherine's question? What is missing? A, B, or C? No one wants to give an answer. Yes, we have a brave answer.
Afrodita, I think all of the above, the identity test, and you should refer to the analytical procedure, and to the microbial examination test, you should refer to the procedure in the Pharmacopoeia. Thank you.
Yep, okay. So that we have got a shout of "we agree" from one side of the room. Online, any thoughts online? Anyone is brave enough to turn their camera on and give a view? I'm looking at Clem and Judith because they can see who's online. I can't. So they're shaking the head. So Catherine, over to you.
So,
Up, yep.
yes, colleagues say all three tests is important. Identity test, reference to analytical procedure, reference to pharmacobiological examination test. So all three be part, and it's missing in total.
Fantastic. Catherine. Yep.
Yeah.
And Céline Thierry said the same thing. So are they right, Catherine?
Yeah, all of them are right.
It's a trick question.
Thank you. Thank you for those responses. And yeah, but this is something we are quite often faced with. Just a very tiny specification for the active substance like this, and yeah, when it's when there are so few parameters stated, it's always a bit suspicious. But yeah, it's true. We need an identity test and a reference to analytical procedures, of course, and yeah, which means a proper reference. And the reference for microbial examination tests according to the LBP general monograph, meaning that we need microbial enumeration tests according to 2336 and tests for specific microorganisms 2638. Sorry.
Perfect.
For the confusion.
Okay, Catherine, I'm conscious that we have 15 minutes left. And how many? Yeah. I think you've got one more slide, haven't you?
Yeah, okay, it's just to mention this here. Other issues that are often related to LBP approval. As I already mentioned, the application is in many cases according to well-established use. According to Article 10A 2001/83 EC. That means that the applicant relies on long clinical experience with the product and refers to literature data. And the limitation here is or can be the proof of comparability of actual LBP, which is described in the submission to the LBP used, which is published in the clinical trials. And I think this is the last slide, the summary. So as take-home messages, we have a European Pharmacopoeia monograph On LBP, it's a general monograph which reflects the products authorized in Europe at the time of setting up the monograph, and it defines the minimum requirements on strain characterization, manufacturing process description and controls, and testing. Also on labeling, which should be done, the potency should be stated in by stating CFU, meaning colony forming units per mass or volume, or alternatively the number of viable cells. And reference is made for microbial purity tests to 2636 and 2638. These are needed for the microbial examination of LBP, and as it's really difficult to do microbial purity tests in presence of millions of viable microorganisms which are forming, which are in fact the LBP. Proposals are described in those texts. How to test the microbial purity for particular species used as LBP, and it's also advice given how to proceed if these procedures are not suitable. And I think this was it. And I want to thank you for your attention, and I'm here for any questions you may have. I'll try to answer them.
That was a marathon. Thank you very much for that. Do we have questions in the room? You'll see. I don't know if you can see that, Katrin. There are some applauding hands going up. I can see who they're from some people within the network. So these are your counterparts in the network who are applauding you. So congratulations, colleagues. Bosnia-Herzegovina.
Just to close case with pellets regarding specification of parameters for freeze-dried pellets. Why only a short list of parameters? Why missing? I know my microbiological parameter is part of puzzle. But why not also other parameters, for example, assay or impurity or related compounds?
Yeah, related compounds in terms of LBP is quite difficult because the only other compound you may have are non-viable microorganisms, so to say, but these are, it's nearly impossible to count them. So you have in the finished product the viable ones together with media components, and to have a consistent quality in this respect, the manufacturing process needs to be adequately described, and you don't have the usual substance-related impurities you see for chemically defined products, for example. You don't have.
It is example, I know that, but because it is technological complex process, pelleting, and it's sure important parameter, assay or uniformity of content.
Yeah, assay is the number of is the potency, is the number of viable microorganisms, because the number of viable microorganisms in the end describes the potency of the product. So you it is the same, so to say.
And uniformity of content also.
Uniformity of content, yeah, it depends on the dosage form you may have. Yeah, the difficulty here might be that you state the content in these are usually millions of cells per let's say per capsule or so, and the viability of the cells decreases during storage. So we have quite some cases where significantly higher number of viable cells is present at time of drug product release, and during stability to ensure that at least stability, a certain stability can be achieved, meaning or a certain shelf life can be assigned with a minimum number of viable cells at the end of shelf life. So yeah, uniformity of dosage. I'm not sure if I have seen that already. Yeah, but you see the test, the assay is, since it's a biological assay, it's quite a variable method, and it may be difficult to establish really uniformity of dosage units here.
Thank you. I'm looking at the colleague who asked the question. Are you content? Are you happy with that?
Yeah, it's okay. Thank you.
Okay, so it's interesting. Any other questions? Oh my gosh, hello. Yes, Katrin, you've got more questions than anybody else so far.
I'm Jelena Letica from ALIMS, and I'm interested in validation of live biotherapeutics. So if you can say something more about it, method validation.
Method validation.
Yes.
Yeah, it's you mean the analytical methods used for release, or
yes,
what
yes.
are you referring to? Yeah, it's described in the end. It's a method validation according to Q2, I would say. You also have to demonstrate specificity,
Okay.
to a certain extent accuracy and precision, which is clearly more difficult because it's
It's about live biotherapeutics. Yes, we know, but how we can validate assay, for example, of live biotherapeutics.
Yeah, you execute. You describe the assay properly, and then you can do things like repeatability.
Yes.
And this is done also for those types of methods.
Okay. Like, for example, for chemical substances, chemical therapeutics. What's the difference between them?
Yeah, okay. Yeah, the difference is, limit of detection, limit of quantitation is not needed, I would say, because but this is also not needed for quantification methods, or for the assay method, for example, when you don't do when it's not an impurity method but a method used for quantification, you need to demonstrate that that your range is, let's say, within 50 to 150% accurate and precise, and similar considerations apply also for enumeration of the coloniform units. So you do repeatability, you do also, which is really important here, specificity, that you show that you count only your product, and specificity is of course even more important when it comes to the identity test. You have to show that your identity test is specific, and this needs to be demonstrated in comparison to similar strains that are handled at the same site, for example. And very often for identity tests also molecular genetic methods are used, for example 16SR sequencing or something like that, to ensure that you are dealing with the right strain. What else comes to my mind? So you have to, the applicants have to select the validation parameters according to the intended purpose of the method. So if it's for identity testing, specificity is very important. If it comes to assay, everything which is related to quantification is important, meaning accuracy and precision, which is done by repeatability, I would say.
Okay, thank you.
So Catherine, before we allow everybody to go and grab some lunch, we have a question online from Céline Thierry. Oh, and another question in the room as well. So let's try and keep the answers concise. So Céline first, and then the colleagues in Montenegro.
Thank you. So I'm Céline from the Belgian agency, and I have a question about the viral safety of LBPs. I am wondering, when LBP strains are isolated from human beings, does FIR 517 on viral safety apply also to the seed lot? Or can we take for granted that human viruses cannot grow into bacteria?
The latter.
Yeah, so we do not care about viral testing in the seed lots.
No. It's it has to be established perhaps right in the beginning, but it's more applicable to I would say to media components, but usually we don't see media components from human or animal origin in the commercial manufacture of LBP.
Thank you.
Question in the room.
Okay. I'm Gordana Stanojevic from Institute for Medicines and Medical Devices of Montenegro. First of all, thank you very much for this very interesting and helpful, useful presentation. I was wondering, you mentioned on one of the previous slides LBP as well-established use applications. So what would be, for example, could you give us some examples of the ways or methods according to which manufacturer can convince us that their product would actually be similar or would do the same as that product that is described in the literature submitted to us?
Yeah, this is always quite difficult. It's the easiest way would be if you have a number in a official strain depository like AMCC or something like that, and if it's clear that the same strain was used in the clinical trial which is described in the publication. So it's this is the easiest way, and by other means it's really difficult. Sometimes you have products on the market which have a long history being used as medical device in the past. So then you have, and if you don't have a manufacturer's change, for example, they can demonstrate that they have used this this type of, or that they have produced this strain and have produced it in a similar way for the last period of time, usually decades, these are the I would say the only possibilities really to relate back to literature. And this is quite a difficulty. It's if this is not ensured, you cannot use the literature data to prove clinical evidence. And also for there are many indications where it is quite difficult to prove really efficacy of the treatment.
Thank you very much. My gosh, I actually did not think we would actually use all the time right up until lunchtime, but we've actually gone over. So, Katrina, that was fascinating. Clearly, you create a lot of interest in the room, and it was actually interesting when we did the questions to hear the groups murmuring and actually having those discussions. So you clearly created some sort of quite a lot of interest.